We cover a lot of complex topics in genetics, and I would say the most complex topic that we cover is regulation of gene expression. We won't do it justice in this class. We won't cover nearly the breadth that it deserves for truly understanding how cells are differentiated and how bacteria respond to their environment. But we will scratch the surface a little bit, and we're going to start that by we're looking at prokaryotes and using prokaryotes as we usually do as a model for the type of processes that happen in eukaryotes. So this video is going to be a lot of vocabulary words, and it's going to be a lot of things that probably are a bit confusing. I would suggest, like many other things, starting with flashcards, but don't just memorize the definitions. Really see if you can explain what's going on in these different types of regulations. So remember that when we talked about our structure of a gene, we talked about there being a promoter at the beginning, and then we had our coding region, and then we had a termination sequence. And this is roughly the structure that we're going to see in all of our genes. But bacteria use something special called an operon. And in an operon, there can be multiple coding regions multiple coded genes within that coding region, all regulated by that same promoter. And so if we look at look at this as if it were a coding region instead of separate genes, we would have our transcription factors come in and bind to our promoter region, and this allows our RNA polymerase to come in and begin the transcription of this molecule. The same thing happens in an operon, except that we have multiple structural genes or multiple coding genes, all controlled by the same promoter. And so what we can get is 3 genes expressed at once. We have an additional feature called an operator. In the operator, this is going to be where we truly control if our genes are turned on or not. And that operator sequence will be found ahead of those genes near the promoter region. So we've already learned that promoter is where our transcription factor binds. And then, excuse me, we have also these sets of regulator genes. Now remember, this is a DNA sequence that produces usually some sort of protein, and the regulator gene is often not part of the operon. So it's a secondary gene somewhere else in the genome with its own promoter and its own region that is the regulator gene. And I've run out of room for my terminator, but it's there. And so what you get essentially is the product of this gene working at the operator or the promoter of a second gene. And it's a really great way to sort of save time and save space by limiting the number of messenger RNAs that you have to produce individually. So you can have one RNA polymerase come in, bind the promoter, stimulated by this regulatory gene, and produce several all at once. In our textbook image for the operon, we have our promoter region, which is where our RNA polymerase is going to bind. You can see next to it that we have the operator. Now, the operator is the location of the regulatory gene product binding. So we have that second gene, that regulator gene. That regulator gene is transcribed and translated into a regulator protein. Now, this regulator protein, the second protein's job is to bind to this operator. And when this regulator binds, it controls gene expression. And this is where that positive negative control comes in. It can act in a variety of different ways. But whatever it's doing, it's binding to that operator to control whether or not these structural genes in our operon are turned on. And we'll talk about the positive negative control in just a few minutes. But let's say that whatever happens, happens, and this operator allows these genes to be expressed. So then they are transcribed into messenger RNA, and then they are translated into their individual gene products. And then those individual gene products can participate in their own biochemical pathway. Now, these structural genes are usually related. So they're doing something within the same or very similar biochemical pathway. So for example, they might each be regulating a step in the same biochemical pathway. So they're not completely unrelated genes or genes that have nothing to do with each other. They're genes that are all going to be used together, which is why they can be controlled by that same promoter and same operator. So this is where it gets a bit tricky, and I'm going to save specific examples for a minute. I realize there's a picture down at the bottom, but we're going to talk about this in more detail with our textbook images. You can use this one to help think through the process if you would like, but for now I'm just going to leave it here. Okay, remember that we have genes that are under positive control, and in a positive control, this is where our regulatory elements stimulate expression. And under negative control, our regulatory elements inhibit that gene expression. So I said we were going to ignore the picture. Let's go ahead and look at it. Okay, so under positive control, we need our regulatory element, which happens to be this little Pac-Man-like molecule here, to stimulate our expression. And so it does that by binding to the operator, which causes transcription to take place. In the bottom, we have a negative control, which means that our little Pac-Man molecule here is blocking expression from happening. So when it is released, now our transcription factors can get to our sequence because our regulatory elements have been released from the operator, which allows transcription to happen. On top of that, some operons are inducible, which means they are usually off but need to be turned on. Others are normally on but need to be turned off. So something can be under positive control and inducible, negative control and inducible, positive and repressible, negative and repressible. Now we are only going to talk about one example. This gets very confusing very, very quickly. So we are going to define what these are, but we're only going to talk about how one example works, partly for the time we have remaining in our course, and partly because this very quickly starts to get outside of the scope of genetics, and our goal here is to just basically learn about gene regulation, not to necessarily talk about every single type of regulation at the moment. we're going to focus on some other things in our course. So let's start with these negative inducible operons. In a negatively inducible operon, we have our promoter region and our operator. And in this case, we have our regulatory element bound to the operator. This is going to block transcription from happening. So we are going to keep our gene off until it is ready to be turned on, in which case another little molecule, our inducer, binds to it and causes that operator to release. And when that happens, we are no longer blocked and transcription can take place. So many of our operons are inducible, which means their default is off and they are turned on. And so in this example here, we have an activator protein. This activator protein is sort of naturally or normally bound to that operator. So it's going to block transcription from happening. In this case, we have a negative inducible operon. Inducible means that we can turn it on. Negative means that our regulatory elements inhibit our gene expression. So we've got our regulator protein that is blocking gene expression, and we want to turn it on. So in order to turn a gene on, an inducer molecule has to be present. So when that molecule is there, maybe it is a little bit of the reactant in a metabolic reaction or something like that, it's going to bind to that activator. And because that activator is now bound to this inducer, this little gene product from somewhere else, or maybe something from the environment, it forces the operator to let go. So when it is bound to the operator, that regulatory protein is holding on, but when the inducer binds to it, it forces a shape change, which causes it to release the operator. And then once it releases the operator, then RNA polymerase can come in, bind to that promoter region, and then begin transcription and then translation. So as the RNA polymerase goes through the operator, it produces each of those genes. We can also have a repressible operon. This is one that is normally on, but needs to be turned off as part of its regulator. Remember that in negative control, our operator is inhibiting gene expression. So in negative control, our regulatory element is going to act to turn off this gene expression. Very often we need a co-repressor, which is kind of like that inducer. It binds to the regulatory molecule, and in this case, helps it bind the operator to block transcription. So it blocks the transcription that is already taking place. So here's our diagram for that negative repressible operon. In a negative repressible, it is already turned on. So repressible, it's on by default. But remember that for negative control, we regulate by binding to our molecule. I'm sorry, our gene, to our operator. So RNA polymerase is working, it's happy, it's bound to its sequence, and it's going to go along and it's going to produce its gene, as long as the repressor stays away. However, if we have a regulator or a co-repressor bind to that regulatory molecule, now it is the right shape to bind to the operator. And when it binds to the operator, RNA polymerase cannot bind. So this molecule here is being blocked by this little operator. And so in this case, we are going to block RNA polymerase from binding. It's repressible, which means we started on and we need to turn off. And we do that by blocking RNA polymerase from binding. Under positive control, our regulatory elements stimulate gene expression. So we call this an activator. The activator is going to bind to the DNA and help stimulate transcription. Now we talked about transcription factors back in our chapter on transcription, and we learned that these transcription factors help to guide our RNA polymerase. So in this case, they are acting as activators. Once we know we need to turn the gene on, then an activator binds to the DNA and encourages transcription to take place.